Welcome back to my YouTube channel where you get plugged with health information as it happens in medical field, including research. Therefore, consider subscribing to get updated about health and health information. Thank you. Today we are looking at the new or the current research in oncologic field about cancer-killing virus injected into human for the first time in new clinical trial. So this is about research where there's a cancer-killing virus that has been identified, which when injected to an animal model showed good results, and it was so much promising for the cancer situation globally. So the aim of this research clinical trial is to evaluate the safety and the tolerability of a novel therapy involving virus that can infect and kill human cancer cells. In this phase 1 clinical trial, a dose of an experimental anti-cancer drug called CF33 HNIS, which is Vaxinia virus, as indicated on the screen, has been administered to the study's first participant. So there's first participant that have been administered this Vaxinia as indicated. This novel therapy involves using an oncolytic virus, a type of virus that can infect and kill cancer cells without harming healthy tissues. Okay. Vaxinia virus is a genetically modified smallpox virus which has been previously shown to be effective against a broad range of cancer in laboratory and in animal models. This clinical trial was conducted by Seat of Hope, a cancer research and a treatment institute in the United States, in collaboration with Imugene, a biotech company in Australia. And they were to test novel oncolytic virus in cancer patients with advanced solid tumors. Based on lab studies, Vaxinia virus may be more effective than previous generation of oncolytic viruses in reducing the size of the tumor and making this therapy especially promising for the global. Okay, now when you talk about oncolytic viruses, what are the oncolytic viruses? Well, these are viruses that are found in nature or are genetically engineered to selectively infect and replicate in tumor cells. As they replicate, they disintegrate and kill infected tumor cells. When the tumor cells burst, they release tumor proteins or antigens which the immune system recognizes as foreign. In this case now, the immune response then elicits against this antigen, resulting in further death of tumor cells. Additionally, the immune system's ability to recognize the tumor cells creates a memory against the tumor antigens, which can help prevent cancer recurrence. Besides providing durable protection, A small dose of oncolytic viruses can be effective against the tumor due to ability of virus to replicate and spread in the tumor cells. Cancer cells express proteins and receptors on their surfaces distinct from the healthy cells that help them evade or avoid the immune system, metastasize, that is spreading of the virus of the cancer, and prevent cell death. Oncolytic viruses use these cancer cell specific proteins and receptors to target them. And these proteins targeted by oncolytic viruses are often common to a broad range of cancers, making these viruses a versatile tool in cancer or in broad range of cancers. Now, how is this Vaxinia virus, oncolytic virus, used to target tumor cells? So, here we look at now what is this Vaxinia virus. Okay, Vaxinia virus, the coded name is CF33-HNIS or Vaxinia, as indicated on the screen, was developed by researchers at City of Hope. Is a genetically modified version of a Vaxinia virus, the spellings are on the screen, which belongs to poxvirus family and is used to make the smallpox vaccine. The researchers have designed Vaxinia virus to enhance its ability to replicate in tumor cells, facilitating a large immune response against the tumor cells. As we said earlier, this virus replicates inside the tumor cells and leading to now disintegration of the tumor cells, releasing the tumor proteins, which now alerts the immune system to come in to destroy them. In addition to this, modified Vaxinia virus also expresses a protein called human sodium iodide importer. That is what we are calling HNIS, human sodium iodide importer, which transports iodide ions into the cells. Thus, tumor cells infected by virus express HNIS, which is human sodium iodide importer, allowing radioactive iodine uptake. Therefore, imaging techniques such as positron emission tomography (PET) scans can then be used along with the radio-labeled iodine as a dye to help track the distribution of virus in the body and its effectiveness. Human sodium iodide importer can also help selectively target tumor cells that accumulate radioactive iodine using radiotherapy. Therefore, upon the successful demonstration of this Vaxinia safety and tolerability, that is the aim of this research. The researchers are also intending to test treating tumor cells using a combination of this oncolytic virus therapy and another type of cancer therapy called pembrolizumab. Pembrolizumab spellings are on the screen. An immune checkpoint inhibitor. When talk of immune checkpoint inhibitors, when talk about the checkpoint proteins, these are the proteins that are expressed by the tumor cells and can cross-link with the immune cells like T cell now. So when we have the checkpoint inhibitor, their work is to prevent or block checkpoint proteins from binding with their partner proteins. Partner proteins can be either on the T cells of the immune system or the tumor cells. So once there's a blocking of checkpoint protein from binding to their partner proteins, this prevents off signal from being sent, allowing the T cell to killing cancer cells. Thank you very much for watching. Consider subscribing if you like to get more information about health. More is coming. We have just started, and I'm going to give information that you will not get it at ease, ensuring that you be updated as a medical doctor, as a nurse, or as a person who is interested in improving the quality of life through knowing what to do and knowing whatever is revolving in health sector. Thank you so much, and may God bless you.